Good morning. Are we connected?
Yes, sir. It's morning, sir. Good morning, sir.
Good morning.
Yeah, I think there seems to be some connectivity issue. They are addressing it. We are sorry for the inconvenience. Let's see. Another couple of minutes, we'll be starting the program.
Sure.
Hello, Dr. Swami. This is Nataraja from GKVK.
Oh, hi, Nataraja. How are you? Good morning.
I'm very fine. I'm very happy that we are connected today. And I must thank the university for inviting you for this conference.
Yes, it is my pleasure. It is a rare occasion when we get an opportunity to reconnect with long lost friends. So,
I know.
this is such a great occasion, a postgraduate science week. I think it's ideal for me to come back and meet with
Great.
you. And yes.
And the thing is, there is some connectivity issue. Our dean, Dr. Prakash, is trying to fix it out.
Mm-hmm.
So that's the reason there is a delay, I think. I'm sorry for that, but we'll be starting in a minute.
Yes, not a problem.
Yeah.
And you are doing well? Everything going well with your work?
Yes, Swami. Everything is going on well, and a lot of teaching work these days. Load, plus I have good projects going on. We are busy and occupied and happy.
Yeah, you can't ask for more than that, right?
I know.
Yeah,
I think they are ready. Yeah, I'll request Dr. Prakash, see whether he's ready to initiate the process. Yeah, thank you.
So our dean has joined.
Okay, Dr. Swamynathan?
Yes.
Yeah, okay, we are sorry, extremely sorry for some small inconvenience, maybe a technical snag from our end.
No worries. I have not seen a seminar where there is a little bit of glitch like this somewhere or the other, so don't worry about it.
Okay. Dr. Ganesh Murthy,
Yes, sir.
can you make us
Yes, sir.
video made available? You need to allow us. Because the host has stopped video.
Just a moment, sir.
Please allow all of us. ಮಾತಾಡ್ತೀನಿ ಒಂದ್ಸಿ.
ಇವಾಗ try ಮಾಡಿ, sir.
Okay, now it is okay.
Yes,
Fine.
yes.
Okay. Good morning, everyone. I wish Swami also comes online with this video. Good to see you, Swamynathan. On behalf of the University of Agriculture Sciences, I welcome all the participants of this webinar or web lecture series of the Science Week 2023. Today's chief guest is Dr. Swamynathan, S.K. Swamynathan, Shivalingappa K. Swamynathan. It is my proud privilege to introduce him also. Dr. Swamynathan, this is the Science Week which is going on in the campus since 21st of August. And today is the last day of the Science Week. It is a valedictory program. And all along we had the two lectures from eminent scientists in the morning session as web presentations. Yesterday and day before, the presentations were mainly from the two speakers, Murali Ramachandra and Jay Kumar from US again. And today's presentation is by you. And it is my privilege to give a brief introduction about you to all my student friends and faculty on this particular Science Week day. S.K. Swamynathan, Shivalingappa K. Swamynathan is at present working as professor, Department of Ophthalmology, University of South Florida, Morsani College of Medicine, USA. Maybe when we hear the word ophthalmology, many of my student friends definitely think how this is actually happening, how he can definitely fit into the Science Week program. So a lot of introduction can give a clear background about where he stands, how he is at present. Shivalingappa K. Swamynathan was born in a small village called Abbigire, that is Shimoga, that is from our state, and coming from a family of teachers, all his brothers, sisters, and uncles, mainly as teachers. He had early access to almost all the biographies of eminent scientists in the world, which mainly really helped him to trigger a lot of curiosity in the science. No doubt, I need to mention he happens to be my classmate, batchmate from 83, 87. He did his undergraduate program from US Bangalore. And after his graduation, he moved to IRI for his master degree program. That is molecular biology and biotechnology at IRI New Delhi. And from there, he went to CCMB, that is Center for Cellular and Molecular Biology, Hyderabad, for his doctorate program. And very interesting thing to mention is that he also got married to our batchmate, Dr. Sudha, happens to be alumnus of our university. She also did her graduation from US Bangalore, master degree from the Department of Crop Physiology, and both of them are working at USA together in the interesting field, which I need to say. He got two postdocs specializing in the functions of Y-box proteins and eye development. This is the major breakthrough for his shifting from agriculture to human science, mainly to eyes, that is ophthalmology. So which is just like traveling from the Earth to Moon, what we all witnessed yesterday, the Chandrayaan-3. So traveling from Earth to Moon, so shifting from agriculture to entirely human science. So the best example for today's talk, maybe as science has no boundaries, and he is really, really witnessing in this regard. So he left India when he won a research fellowship at the University of Missouri, Columbia, that is in the Department of Molecular Biology and Immunology, USA. And later he first started working as intramural research training associate, and then as a staff scientist in 2005, and later as associate professor, and also as a director of tissue culture and histology core module, that is Department of Ophthalmology, University of Pittsburgh. That is during 2020 to 2022, he served as a director there. At present, he is working as full-time professor in the Department of Ophthalmology, that is University of South Florida, that is Morsani College of Medicine, USA. Very happy to mention that he is bestowed with many prestigious awards. Apart from his many fellowships, awards during your early education, he is bestowed with many awards, including National Eye Institute Career Development Award in 2011, Silver Fellow from Association for Research in Vision and Ophthalmology in 2022. And one of the interesting award, and which is really noteworthy to mention in front of all my students and faculty, is that he was honored with Asian Scientist Award, which is the renowned award for the Asian research scientists. Swamynathan handled many research projects, and ongoing work in the Swamynathan's lab itself is supported by FARVO grant, which is a very prestigious grant from USA, that is in the National Eye Institute, NIH. And his inventions are extensive, no doubt. So he has got 2 US patents and published 46 full-length research articles, 31 research abstracts, and that is actually in the peer-reviewed journals, and including 1 chapter. And he has delivered 40 seminars and lectures. Apart from these, he also serves as the member of the editorial board for several journals. Today's guest speaker at Science Week 2023, Dr. Swamynathan, is truly a great scientist, no doubt. So it is a great asset to the scientific community also. I'm so happy that Swamynathan is here to address all my student friends and faculty in the Science Week. Swamynathan, so we're all honored to have you here for the Science Week celebration. Hope our joyful multitude increases with your talk, and our students will definitely enrich with your well-nourished and acknowledged knowledge what you all gained all along, and you will be a model for us to show and excel in the field of human science, shifting from agriculture, and how the journey is. You can really give us a lot of information in this regard. And once again, I need to thank all the officers here who are all joined here online, and my faculty, professors, associate professors, and all those who are involved at this moment, and all my postgraduate students. And another important thing which I need to mention it to you is that, so during this particular Science Week program, 346 posters have been presented, and 96 oral presentations were made by our PhD graduating students, PhD scholars. And before their completion, so this particular event need to happen. And the event is actually happening in the 9th in this series. Since 9 years ago, we started this one, and a lot of innovation, transformation happened. And this is how the development of postgraduate studies, which is existing in the university, is functioning. And on behalf of the university and my behalf, I need to welcome you once again and to deliver this address. And I thank Dean PGS Postgraduate Studies for giving the opportunity to introduce you also. So now the time is yours, and the platform is yours, Dr. Swamynathan. You can give your presentation, share your slides, and of course, the people from Dean PGS will coordinate the rest of the things in this regard. Thank you.
Thank you very much for a very kind introduction, Dr. Prakash. It is an honor to be here to present this talk on the occasion of this PG Science Week, which seems like a wonderful opportunity for the students to present their ongoing work and receive feedback from the other students and faculty. And before I begin, let me give a big shout out to ISRO scientists for the success in Chandrayaan-3. It's really a proud moment for all of us, and all through the day I've been walking with my chest held high, and I'm really happy. Anybody who I met, I was asking, "Oh, did you hear this? Did you hear this?" and so on. So that must have come up as a topic of conversation in almost every interaction for you also today. So with that, I also must mention before I move on to the actual talk that Dr. Prakash is actually a very close friend of mine. Just to give you a little bit how to show how close we were. we actually went swimming in an open well in his village when we were college students. So there are many other advantages which I won't get to, but right now I think that is sufficient, right? So let's go to the presentation mode. That is what I'm going to share.
You need to share.
Yes.
Okay.
Yes. So do you have the presentation in a slideshow mode now?
Sir, it is
You need to make it as presentation mode.
That is what I'm trying to see. Do you have it now?
Yes.
Yes. Okay, good. So I was asked to give a talk about science for society. And I started thinking, there is a lot to be said, and many deep thinkers have said a lot of different things about it. And what is it that I'm going to tell you from students from Agricultural University and being an alumnus of the University of Agricultural Sciences? How do I go about talking for this? Was an interesting challenge. So before I get into this, at the outset, I must recognize that I am who I am today because of my parents, my siblings, my friends, my teachers, and it was their unconditional love and the many words of wisdom, encouragement, which really propelled me to where I am. So I want to take this opportunity to thank them for every word of their encouragement, every act of their kindness, and every moment of their unconditional love. Now, Prakash already informed you about my journey through living, being born in a small village to my parents as a seventh child, and I had a really wild childhood. It's a miracle that I survived through that, and I came out and went to college. And in high school, I really became serious about studies. Till then, I was really a wild child. And once I got serious, I really got serious about what I wanted to do. And that was the time when I began taking interest in sciences and the discoveries of life-changing nature. They really made a deep impression in my mind. And I decided to go after college, after PUC, I decided to join the University of Agricultural Sciences because I was really drawn by the campus, the beautiful campus. Though I had the opportunity to go to any choice of any agriculture, any engineering branch in any college in the state, I chose to leave all that and come to GKVK. And it was a good thing I did, because that's where I met Sudha, my future wife then, and also made many lasting friends whom I even today I continue to have a close friendship with. After graduating in 1987 with a BSc in agriculture, I went for a short while to Dharwad to study plant breeding and genetics. But then I got admission in IIRI for masters in molecular biology and biotechnology. So I decided that because of my deep interest in genetics and molecular biology, I decided to take that up, and I went there. And 2 years later, I got admission in the Center for Cellular and Molecular Biology in Hyderabad. That's the CSIR Institute. And that is where I made the shift from studying rhizobial symbiosis in legumes in masters to studying animal genetics and animal gene regulation and so on during my PhD. And even as a grad student at CCMB, I got married to Sudha, which I tell you has been a blessing. We have a child together. Our son Gautam is now 28 years and he's working in energy sector. And soon after my graduation, along with Sudha and Gautam, we moved to the University of Missouri and then to the National Eye Institute, where I started working on the eye development and gene expression. And this was all possible because of the fact that deep down, genetics is the same whether you take it from whether you study it from the perspective of plants or animals. The way basic biology works is the same. So that gave me a solid confidence in making this shift from studying agriculture and plant breeding to studying human or other animal genetics and gene regulation and so on. While working at the Laboratory of Molecular and Developmental Biology at the National Eye Institute, I got the Career Development Award, and with that I went to the University of Pittsburgh, where I started my independent career. That was at 2007. So now I graduated out of the University of Agricultural Sciences in 1987, and it took me 20 years to come to a stage where I was becoming independent scientist. So that is how long it would take with a PhD and postdoctoral fellowship and so on. It was really a journey that we had to persevere through. But once I joined in the University of Pittsburgh, I started the Laboratory of Ocular Surface Development and Gene Expression there. And in 2015, I got the tenure and promotion. And in 2023, we moved here to the University of South Florida, where I'm continuing to work as a professor. Now, throughout my independent phase of my career, Sudha has been working with me. She has been a solid research associate and has been instrumental in developing one of the two projects that I will briefly introduce you in the next few slides. So we study the ocular surface. Ocular surface meaning the front of the eye. You can see that whenever you look at the mirror, you see that there is the clear part in the center and there is a white part that surrounds your eye. And that the clear part is what is called as the cornea. And cornea is surrounded by this conjunctiva on the sides. And this conjunctiva actually goes in under the eyelids and folds back and comes up to become the under part of the eyelids. And it is all connected. And the connected surface is lined by epithelial tissue. Now, the lacrimal glands that are located right above the eyelids in humans secrete bulk of the tear that the tear fluid that covers the front of the eye. Now, the cornea that is transparent here is more or less the same structure as the skin that is covering the rest of our body. But you recognize the importance of cornea, that it has to retain its transparency for light to go through, because light passes through the cornea and then through the lens and then only falls on the retina, which is on the back, shown here on this section on the right side. So what happens is, because of the difference in refractive index between air and the cornea, and also because of the curvature, there is bulk of this refraction that occurs right at this junction, the air to cornea interface. And the rest of it is only fine tuning to focus on the fovea and the back of the retina. So this really acts as a window to the eye, and it is important that it remain its transparent status. And how that is achieved, it is to retain its transparency, yet it has to almost function equivalent to the skin in the rest of the body, where skin defends against the external elements, right? Water falls on it, it just runs off, and it keeps the outside out and inside in. How that is achieved by the cornea is a big question. And another big interesting thing is this is actually cellular structure. There are 3 different distinct cellular layers within the cornea, and all those cells have to survive without any blood vessels growing in there, because blood vessels, if they grow into the cornea, they will block light, right? That becomes an impediment for our vision. So we cannot see properly if there are blood vessels. So there are active mechanisms to keep our blood vessels from entering the cornea. They are blocked from entering there. How that is achieved is a question that we have been very interested in for a long time now. The projects, coming to the specific projects that we are working on, I want to tell you a little bit about the very first project, that is something called ocular surface homeostasis, meaning how the once developed, how does this ocular surface remain in its state as without being disturbed or without changing it. So I told you the surface is covered by this cell type called epithelium, and there are transcription factors, very similar function as in plants, transcription factors that regulate gene expression. There are many different transcription factors, and these KLF4 and KLF5, Kruppel-like transcription factors. They are 2 of the most abundant transcription factors in the cornea, and they are very interesting in the sense they have identical DNA binding domains, yet they have different functions. And identical DNA binding domains and different functions can be achieved because of the difference in their regulatory regions. Now, why do cells require that kind of a setup? We started thinking in terms of their functions and in terms of their expression. What you can notice is that KLF4 is expressed, shown here in red color. It is expressed in almost all the cell layers of the epithelium, whereas KLF5 is expressed mostly in the basal cell layers. And that is important because KLF4 is known to be a pro-differentiation factor, whereas KLF5 is known to be a pro-proliferation factor. And so we immediately hypothesized that these cells have, whenever a cell has a choice, it has to decide whether to divide or to differentiate. What determines the outcome of that decision is this relative balance in the activity of KLF4 and KLF5. So this hypothesis that the choice between corneal epithelial cell proliferation and differentiation depends on the balance between anti and pro-proliferative activities of KLF4 and KLF5 respectively was largely proven by our lab over the last 15 years. It's painstaking, detailed work that resulted in about 15 publications. And these publications have gone on methodically to show how the functions of KLF4 and KLF5 are crucial for maintenance of the corneal epithelium. And in a second project that we are working on, I alluded to this problem, that is, the cornea has to remain transparent, and yet it has to function like skin that is covering the rest of the body in terms of the barrier function, in terms of keeping the outside out, in terms of defending against bacterial pathogens or viral pathogens or dust or cosmetics or anything else that we put in our eyes. Now, what you need to appreciate is that there is a mechanism of inflammation. You all have experienced a bug bite. When a mosquito bites, you will see swelling, pain, and reddening, and so on. And that happens because the body responds to the toxic chemicals that are injected there by the mosquito in the process of its own feeding by sending cells that defend against that toxins, to clear away the potential for infection and so on. So all the cells, when suddenly they rush to the bite site, they result in swelling and pain and reddening and so on. The problem with cornea is for it to be transparent, it cannot swell. If it swells, immediately you start having blurry vision because proper transparency is lost. When the light goes through that, it starts getting scattered if there is any swelling, and that needs to be avoided. That is, if there is any minor insult, like a small amount of soap goes into your eye or dust particle falls, and things like that, you cannot keep mounting inflammation to everyday minor insults. On the other hand, if there is a severe infection, say a viral infection or a bacterial infection, it needs to be cleared. Otherwise, it is much more dangerous. It can have the potential to make you blind. And when that happens, the body needs to know that there is a need for inflammation, and that has to be taken care of. And how this sort of a decision is made, when to mount inflammation and when not to mount inflammation, is a very interesting question. And we have been working on this again for the last 15 years or so. And our work has identified a new protein in the tear fluid. That protein is called SLURP1. This SLURP1, SLURP1, it's a long name, so it's better to stick with the acronym here. It stands for secreted Ly6 related protein 1. So this is a small peptide that is present in the tear fluid, and it helps in telling or signaling to the immune system that everything is fine, there is no need to mount inflammation. But when there is a pro-inflammatory cytokines and other insults resulting in oxidative stress and consequently reactive oxygen species are present, the expression of SLURP1 is shut down. And it immediately signals to the immune system that when SLURP1 is going down, there is a need for inflammation, and that results in further development of inflammation. And this is a project that Sudha has been deeply involved in over the last 12, 13 years and is spearheading. Again, we are the only lab in the whole world working on this project in the eye. And we have this pride that we have made a difference in our understanding of how the ocular surface homeostasis is maintained by Kruppel-like factors scale of 4 and scale of 5, and how immunomodulation of the ocular surface is achieved by this protein called SLURP1. So with that as the introduction to our work, I want to talk a little bit about the main topic for today, that is science for society. Right? Before I get into science for society, I want to tell you, briefly, if you are wondering as to why or how this type of a transition for me was possible from studying agriculture and plant breeding and genetics to come into the School of Medicine, where I am teaching medical students and studying human biology. It's all possible because of the nature of our agriculture course. It's a beautifully designed course with, which teaches you a lot of different topics, varied subjects such as economics, psychology, sociology, meteorology, biochemistry, genetics, pathology, entomology, agronomy, plant breeding. You think of a topic, it is there. There is a course in that in agriculture college. And that really gave me a broad background and a very solid foundation in my early education. And that helped as I went on to study other things. So if there is any discussion on any topic, I seem to think that, oh, maybe I know something about it. That is thanks to agriculture college. And, the best thing about it, it was practically free because it was heavily subsidized by the state government. And there you go. That is the first exposure that I had of the impact of society on science. The recognition by the government of the importance of this kind of an education and the need for it to be subsidized, it means a lot. And this is what promoted, not only me, but many prominent people who have come out of our agricultural college. You will keep hearing about it in the news almost day in and day out there. So with that, let me get into this science for society as the topic for the rest of the discussion. Now, we all recognize that science is a collective endeavor, right? People have to work together to make solid progress in any field, especially so in science. Science helps provide for our basic needs, right? Food, shelter, health, energy, and so on and so forth. And it also makes life worth living, life a lot more fun, entertaining, and it provides us high quality music, high quality entertainment, and this type of a communication. Just imagine how I am sitting here in Tampa in the middle of the night and I'm talking with you, thousands of miles away. It's a face-to-face interaction. How is such a thing possible? It's mainly because of science, and this science was is responsible for changing our society. This kind of progress, what it does is it nourishes our soul, our spirit. When we understand the cause and effect relationships, in myriads of our daily interactions, what we feel is a sense of security. We feel a sense of accomplishment, and this is a big deal in improving the quality of life for the society as such. And another thing that science does for society is that it frees up time for humanity to engage in cultural activities. It develops cultures. Scientifically advanced nations also are culturally advanced nations. It's really important to recognize that. And its role in creating new knowledge and in improving education, improving quality of our lives, it's really paramount. Now, society in turn has to support science, right? Otherwise, scientists will find it really difficult to thrive in the absence of support from the society. So this societal support is absolutely important. So you immediately recognize that science and society need each other, right? It is imperative that they work together, correct? Science has to respond to societal needs and global challenges, and the society must popularize science and promote citizen participation and so on, and promote education and so on. To face this sustainable developmental challenges, governments and citizens alike must understand the language of science. It's important that they speak the same language or similar, understand each other. And our policymakers have to understand the problems that are faced by the society, and also how the scientists are trying to solve them and what the scientists need to do that, to do that efficaciously. And this type of an arrangement is the ideal. Do we have it every place, every society is a different story. Many places have been encouraging science for a long time, and there are periods of highs and lows in any society if you think about it. So we next look at what the scientific process itself is, right? And how society influences it. Scientific process, you probably have read about it in your experimental biology courses in the final year of agriculture, where you have to take a project, and essentially what it involves is careful analysis of literature, right? You have to study what is already known about a particular topic and make also observation. You study and observe a phenomenon, and based on that, putting those two things together, you form a hypothesis. And once you make a hypothesis, ideally that's a testable hypothesis. Once you make this testable hypothesis, you test that, and you come up with an answer whether your prediction was correct or not. And that answer in turn opens up new questions. And so this process keeps on happening. It goes on forever. That's essentially the scientific process. And at the end of each of these, or at the end of specific set of such repetitions, you may want to publish your results so that you invite the rest of the scientific community to study your findings and either criticize or support and praise, depending on how the society looks at, scientific society looks at it. So that is important. But now this is the basic scientific process, but the science itself can be broadly divided into curiosity-driven basic science research or application-oriented translation research, right? And this translational research or basic science research, they both involve the same scientific process. Now, ultimately, what you want to happen is they both lead to this commercial benefits, so that there is society that is benefiting, or the human beings or your community ultimately reaps the benefits, and in turn that supports the science, and there is this kind of a feedback that occurs between societal needs and commercial benefits that are driven by either curiosity-driven research or application-oriented research. And so this type of an interaction keeps on happening, and this is pervasive. Wherever you look in science, you see that. So we talked about the mutual dependence of science and society. Now, are the outcomes of this mutual dependence always predictable? Not necessarily, right? There are unpredictable outcomes. The best example that we can think about, are the most recent one, instead of, let's not call it the best. Probably the most recent example is COVID-19. Over the last 4 years, countries around the world provided massive support for science that is aimed at research in COVID-19, right? And this COVID-related research not only improved our understanding of viral infections and how the virus life cycle is in the human cells and so on, but it also helped rapid development of mRNA vaccines. Now, this mRNA vaccines that are developed as a result of this COVID-19, or as in response to COVID-19, the technology was already present, even much before COVID-19 happened, but it was never tested. The technology was present, but the appearance of this pandemic, gave an opportunity for testing this type of a technology. And society's desire to control this pandemic resulted in development of the technology that can, again be useful for other uses in the coming days. Now, that is one example. And then there is another example I can think of, that is one little step taken at the right time, in the right technology, or in the right area, can have a profound effect on the progress in overall science as such. You can think of the solving of DNA structure. How fundamental it was when 1953 Watson and Crick came up with this solution for the DNA structure, how it changed, how rapidly molecular biology developed in the next 40, 50 years. It's out there for everyone to study. And listed here is an example of PCR, a simple technique of polymerase chain reaction where you amplify a particular fragment of DNA. This technology changed the way we do biology. We study biology. It resulted in a lot of new additional technologies and many companies and economic revolutions and so on, all because of just one technique. And that technique is something that people cannot live without today in molecular biology laboratories. Another situation that you can think of is the computer science developments. How one field, major developments in one science can influence another unrelated area. So computer science revolution in the last 20 years or so aided systems biology. Now most of the biology that we are talking of is done at the level of systems, not just one cell. Even if you are talking of one cell, all the genes in that one cell. So it's the whole system as such is addressed. So genomics, proteomics, lipidomics, metabolomics, all this kind of large scale analysis have only been possible because of the revolution in computer science and our computing power that can handle enormous amounts of data at once and synthesize meaningful outcomes from that data. Now, the final thing that I want to bring about in this science and society mutual dependence and how unpredictable the outcomes are is you cannot always depend on this targeted, focused research. You have to keep your mind open. There is always a chance for serendipitous discovery. There is a chance for an attentive mind to catch something that can change the course of our history. Plenty of examples, if you look through, the things that I could see are discovery of penicillin, discovery of X-rays, microwave ovens, and this color mauve. Very interestingly, this organic chemist, was searching for a synthetic alternative for quinine, and in searching for this, he mixed 2 or 3 different compounds and came out with a solution that gave this very novel color that he called mauve. And this color became so famous in Europe. In all the societies, the ladies started wearing the clothes in this color. And in turn, what happened was there was a big demand for organic chemists who could synthesize this color. And that's in turn triggered organic chemistry, support for organic chemistry in Europe. So you never know where things can turn or how things can happen. And there is this example of Viagra, for example. This is a drug that was being developed for cardiac failures. There was pacemakers, again, the similar story. Sticky notes, the 3M sticky notes that we keep sticking all through things like this in our offices. We do pull out one and stick somewhere. Actually, it was a failure because the scientist who was developing that was tasked with a specific requirement for a different kind of a glue, and all he could come up with was this faintly adhesive but repetitive nature that does not stick to the surface, but it helps the material or the paper stick to the surface. Ideal for a sticky note, but not the purpose that he was searching for. So that these are all examples of serendipitous discoveries that really changed the way things work in our day-to-day life, right? So you have to stay attentive, is what I'm trying to tell you. Don't always wait for the outcomes of targeted research, right? So talking about research funding, what is the role of society in supporting scientists? The research funding influences the path of science by encouraging research in specific topics. That's a given. And when it does this, it also unavoidably, it takes away the research focus from other topics. Some of them could be crucial, but the society does not think so. At that particular moment, it weighs the needs of another topic as much more important. So it funds in that area. And funding agencies like, say, DBT or DST or ICAR or other, like within the United States, maybe National Institutes of Health and NSF, National Science Foundation, they can make or break a particular field of science just by their decision to fund or not fund a particular field. Now, the cancer research has been going on for a long time. For at least about 40, 50 years, there has been this cancer-focused research. But there are spikes, depending on how, when the government policies change, spikes in cancer funding, and that in turn can result in big difference in the research focus. Similar thing happened with HIV when there was HIV problem that was becoming unbearable in the US. There was a tremendous boost in research focusing on HIV. And the most recent example, again, for COVID-19, you can hear that for a long time to come, how COVID-19 research facilitated a lot of other things. Now, this need not necessarily be only a government's or funding agency's role. There are foundations, private foundations, or even individuals. There is, for example, the best example that comes to mind is X Prize, that is, huge amount of money offered to tackle a particular problem. So there is this kind of funding that can guide research in a particular direction. Now, societies have changed over time, right? And consequently, science also has changed. The events of the first part of 20th century, around 1915 and again at around 1940, the world wars, the World War I and World War II, they resulted in tremendous changes, big changes in our research focus. That was the period when most of the research funding was targeted towards the war-related research. Defense funding was primarily driving all research. All available scientists were told to only work on defense-related projects and nothing else. So that resulted in the uncovering of nuclear energy that it has its own advantages and disadvantages, but it came at a cost of ignoring other areas of science for which the society paid a price heavily in some cases. So this is something that we need to guard against. Too much influence from one individual or from one society funding agency in a particular area of research is welcome for that particular area, but not so for the rest of the fields of research. So it is a responsibility that the scientists and the science needs to guard against and make sure that the society does not fall into such trap. In talking about that trap, I want to give you this quotation by a scientist by name Donald Hart. He was a former director of NASA Research Center. So what he said was, beginning with the plow, science has changed how we live and what we believe. By making life easier, science has given man the chance to pursue societal concerns such as ethics, aesthetics, education, and justice, to create cultures and to improve human conditions. But it has also placed us in the unique position of being able to destroy ourselves. It's true, we are capable of doing enormous harm to humanity if if some of the developments fall into wrong hands. Again, this results in an important question that is related to ethics. We will come to that in a few minutes. Now, you are all in agricultural university. What is it that you can look to for inspiration? In contemplating the topic science for society, where do you draw your inspiration from? Being a scientist or a student in this research agricultural university, you don't have to really look hard or look far. It's just looking at the population, the way it has grown over the last 12,000 years gives you an important indication that the way things are going to be challenging soon. It's staring at our face. The world population, for example, has increased from about 1 billion in around 1800 to around 8 billion today. And this does not show any signs of decreasing. The rate at which population is increasing is not showing any signs of decrease. At the same time, the available natural resources, the size of the earth, arable land, water, all that has not changed. So we definitely we need to make smarter, more efficient use of available resources. And this is where the society is going to need you smart scientists to come up with innovative ideas to feed this population that is ever increasing. Now, if you look at it, the population is not only increasing in numbers, but the number of old people in the population also is increasing because life expectancy is improving as well. This is a very unique problem that the countries are beginning to see now. There is a shift from primarily 20s and 30s as the workforce. Now there is a shift to 40s, 50s, and 60s as the primary workforce in countries. Now with that, what happens is soon there will be a stage where there will be many more old people than there are working age group people. And that is a situation that is going to present totally unexpected kind of situations. First of all, our human body evolved up till around 1800. Our age, our expectation, life expectation was below 40 years, 30, 35. So that's enough for one generation to live and procreate and have the kid, and so that used to be the case. Our evolution did not prepare us for living longer than that. But after that, this expectation, life expectancy has gone up drastically, and this has happened all over the world. It's not necessarily in developed countries, but also in countries like Africa. It's relatively less, no doubt, but the trend is the same, nevertheless. So with this, there is going to be a problem where the human body that is not evolved for living longer is now living longer. With that, we have unique problems that are associated with old age, for which scientists need to come up with answers. The society is going to look to you, scientists, for answers as to how do you solve this problem that is unique to the older population. It's not an ignored population. You cannot ignore them. They're highly productive in their age, and now they have reached a stage because of the advances in science, advances in our living conditions, advances in healthcare, and so on. Our understanding of biology, all that has resulted in this improved life expectancy. So it's not their fault. You cannot ignore this older population, and we need to come up with answers to the problems that are unique to this population. How that is to be done is going to be an interesting question for curious minds to solve. Now, if you see this graph, what you see is that there are two big dips around 1950, late 50s, and also very recently after 2020. So this is where the first dip that we are seeing here, the drop in life expectancy happened because of avian flu, H2N2 flu, that was responsible for millions of people dying all over the world. And this one, you probably already have guessed, the most recent one between 2020 and 23 is the COVID-19. The effect of that is undeniable. You can see that the life expectancy dropped precipitously, and hopefully it will not continue this downward trajectory. It will come back up and go further back. So that is the kind of information that is out there. Now, the question is, what do you do about it? Right? So I started telling you about the ethics, how science, society, and ethics are also interrelated, and how scientists need to consider ethical implications of their work above everything else. Now, the truth is that we live in a world where scientific advances continuously challenge our values. Every new discovery, every new way of thinking, new way of doing things challenges our previous values, right? Our generation challenges the seniors in many ways they live in, and that is based on our experience or our learning or our values. It's not to say that the previous generation had wrong values or wrong customs. It's changing. Change is the nature of things here. So advances in science result in this kind of continuously challenging situation for values, and we have to live and decide based on fundamental values of human dignity. That has to be the primary concern. What is it? It can be slightly different for each of us. The way we define human dignity can be, but deep down, it's the same. You have to value the life, and every person is equal and has the same potential and same ability to do things. Some are blessed in different ways, and some are blessed in different categories, and it does not mean that the others are poorly or challenged in many other aspects, and so on and so forth. But this type of a belief in fundamental human dignity is important in anything that we decide when it comes to science or scientific approach to a particular problem. Even to identify the area of research, you have to think in terms of ethical values associated with it. Scientists have this obligation to try and resolve such ethical conflicts, because they frequently have this, what you call as first-hand information about these dilemmas, much before the society comes to know about such ethical dilemmas. Scientists have this ability because they are the ones who are creating these problems, right? They know what sort of issues can come up with. So they have this first-hand information, and they need to be cautious about the path they are taking. So here what I've done is I've listed just a few for giving you an example of some hot button issues as to what sort of unresolved ethical concerns can be. For example, relationship between public and privately funded research is always an issue. There is what is the right thing to do, and then use of genetically modified crops, whether it is the proper way to go. And the question is, when one is starving, what do you do? If it becomes a question of survival, is that acceptable? There is this ethical questions that need to be discussed, that need to be carefully analyzed. And then, so in animal sciences, embryonic stem cell research, true, it has the potential to come up with cures for severe diseases, but if it means that you are destroying embryos, is that ethical? It's an unresolved issue and there is so much discussion that has already gone on and is continuously going on. Similarly, the research on gene editing in embryos, so you can make custom babies, made to order babies. If you can imagine this, if this is allowed, what sort of an impact it will have, on humanity. So just because the scientists have the ability to edit embryos, does it mean that it should be allowed? These are the questions that the society needs to have strong answers for. And then most recently, the AI technologies such as ChatGPT, how are they going to impact us, and how are they going to make a difference in the way science and society interact? That is going to be a big unknown right now, but hopefully it will be resolved. People are just now waking up to the possibilities of this technology. So these are just some examples, and I'm sure if you think about it, you can come up with your own list of such ethical concerns. So I have spoken at length and touched upon various aspects of the issues related to science and society, and how science, and it's science for society and society also for science. It should be, that's how it should be. But then there are needs of having these checks and balances also for science and society, right? They need to have good checks and balances. So a topic that meets a societal need is often more likely to be supported, I told you, for funding and so on. And is it the role of scientists to make sure that the other fields are not ignored? That's one way of having this type of checks and balance from the scientists. On the other hand, the ethical issues that I talked about just a few minutes ago, those need to be curtailed by the society, and that's so there is going to be a lot of that type of discussion you will, as scientists, encounter, and you will go through in your own mind while choosing a research topic for yourself. And the other question that you need to think about is, does science bear responsibility for misuse of its findings? If some crook comes up with a an idea of misusing, say, the nuclear energy, is that blame squarely falling on the scientists who developed it? Because nuclear energy has also powered millions of homes all over the world. It has given energy for transportation. It has given lots of advances in our understanding, and it has brought us closer to the structure of molecules and atoms and so on. But just because a crook misused this knowledge, does it make the scientists to take the blame? Or if so, how should they have foreseen this type of consequences, and how should they have acted? So this type of a situation calls for the question, does society have a right to impose restrictions on science? And if so, what are those restrictions, and what type of knowledge, and to what extent? So these are all the questions that, there is no one answer to it. I leave you to think about it as I end this talk for today. This has been an honor. I really enjoyed talking with you and reconnecting with old friends from the past. And thank you very much for your patient listening as I went rambling on multitude of topics. Thank you.
Thank you, Swaminathan. Thank you for your nice talk and addressing all of us on the science for society. I think a couple of questions are also there. Let me just ask Dr. Nataraj Karba and Banu Prakash in this regard, and also hear from my student friends and faculty, if at all, if there are any questions.
Sir, you can exit from the slide sharing so that can come on to the view, sir.
Yeah, he is already on view.
Still, slide sharing is there. Slide sharing is there.
No, Banu.
Yeah, sir.
Slides are not visible.
Slides are not visible, but here it's stuck, I think. So in my view, it is coming out. It's anyhow. So thank you very much, sir, for your nice presentation and opening talk that what you have given. So there are few questions which they have posted. So I'll just go through that particular questions and then come on. Yeah. It's coming up again. Okay. The first question is from the Nataraj Karba, Professor Nataraj Karba. It's a specific question which that how KLF TFS regulate ocular surface function, and are they crucial TFs for vision? That's what, yes.
Yes. The answer is answer to the second question. Are they crucial for vision? Yes, they are. How they regulate ocular surface function? More than maybe 10 different ways they do it. Most importantly, what they do is they work together as a team, KLF4 and KLF5, work together as a team. KLF4, what it does is it pulls a cell towards differentiation, whereas KLF5 tells the cell to divide. Now, any cell in the corneal epithelium has this decision to make, whether or not it is dividing or it is going to differentiate. So that decision is extremely well balanced and it is well regulated. At any given time, only a small number of cells can be dividing in the corneal epithelium. If that small number goes out of control, it results in tumors, ocular surface squamous neoplasia, and other kinds of problems. On the other hand, if there is too less of cell proliferation or cell division, that results in very thin corneal epithelium, that results in porous surface, which means that there is no barrier. Everything that is in the environment is sensed by our cornea. You probably have already experienced it. Cornea is the most innervated tissue in the body. It has the most nerve endings. Because of that, it's the most sensitive. Even a small eyelash, if it goes there, it gives you so much problem. For the rest of the day, you won't be peaceful. So that happens because of the nerve endings there. And all those nerve endings can be exposed if the epithelium cells are not there. So these are really important. And that is just one way. And then there is another way. These KLF4 and KLF5 both can regulate the formation and function of cells called goblet cells. Goblet cells are the ones which produce mucin, and these mucins are a part of our tear fluid, and they are important in protecting from bacterial infection. So if the KLF4 or KLF5 is missing, then goblet cells are not forming properly or not functioning properly, and that results in problems.
Okay.
And the way cells behave is very different. There is the basic cell type called mesenchymal cell. Mesenchymal cell is more or less like a tumor cell or a cancer cell. And epithelial cells have this tendency or a ability to go towards this mesenchymal cell fate. KLF4 keeps the corneal epithelial cells in epithelial state. If you take away KLF4, if there is no KLF4, cells then can easily go into mesenchymal state. So that kind of a problem is common. If that happens, it again results in bad outcomes for the cornea. So these are just a few ways in which KLF4 and KLF5 regulate.
Thank you so much. Basically, they are regulatory transcription factors, not downstream proteins.
No. They are regulatory transcription factors which regulate thousands of genes in the cornea.
Great. Yeah, thank you very much.
There's one more question, sir, which addressed like this way. So, now please share your views on the relevance and applicability of humanoids to society and the ethical concerns around them.
So, humanoids definitely no, right? It's ethically not allowed in anybody's mind. I hope so, at least. Unless there is really crooked individual who wants to form an army of strong soldiers who only listen to him and nobody else. So those are the fears of development of such technology. So it's to be avoided any cost. Now, if you are thinking of organoids, that is a different story. Organoids, is a field of study where you can develop specific small organs in culture using cell culture techniques, and that can be useful for understanding biology without harming model system or embryos or other costly methods. So that is definitely acceptable, provided it is well within the ethical means.
And now I call upon our Dean PGS to have few words.
Okay. Thank you, Dr. Swamynathan. Thank you, sir. Sir, I text you a message through WhatsApp requesting you for your delivering the lecture. I think you must be knowing I'm Dean PG, Dr. Prakash here. Of course, your today's talk, sir, very interesting. And from ophthalmologist to you traveled all the way through from society, science and society, science and society and ethics, and our research double-edged swords and the balance. It's a very really thought-provoking, sir. We enjoyed your program. Very nice program, sir. That to our students also. And one question, sir. Majority of our agri graduates who studied here, after going to Western countries, in particularly USA, they embrace a medical field. What may be the reason, sir?
I think it is primarily money, because funding for research, we talked about the influence of society on science. Funding for research in human science outweighs the research funding for plant biology in all over the world, not necessarily in the Western countries. It is because that is where they see the need to be the foremost. And when that happens, scientists who come here quickly realize that there is a better chance for funding in animal science than in plant science. And that can be a factor. Second is there is already a large number of animal science scientists who are working already in this field. So that critical mass of plant biologists is missing in many universities. Not every place that is available. It's in select few agricultural universities or other places that you have plant biology being practiced. So that makes it harder to find a place where you can do this research. So those are the two main reasons that I think drive.
And one more question, sir, regarding ophthalmology. During my say at the age of 25 to 35, I suffered the allergic to the eye, red eyes. Almost it was there persisting for 10 years. I consulted an ophthalmologist and all the other doctors, including allopathic and homeopathic, even Ayurvedic. I did not cure it. But one thing, the local medicine is there. Traditional medicine, I think you must be knowing. Nimbu juice, lemon juice, a drop of it. I put it, it gone away. So far, I have not noticed. Why it is so, sir? Because, but doctors are advising not to practice it. During that time, I put one drop for the both the eyes. It was unbearable pain. It is not able to tolerate. I can tolerate that. Can able to tolerate that severity of the juice, but not this one. Therefore, it has cured. It is not all there with people.
So just
right
one
now.
drop.
And my also safer now.
Just one drop once. That's it.
Yes, sir. One drop of limbu juice.
And only once.
Yes, sir. Only once. It
Wow,
has gone away in another 2 yes.
That is remarkable, and I'll think about it. What could be the reason?
Yes, sir.
How that could have helped.
I think you must be knowing locally in villages, in our villages.
Correct.
Before this medicine comes, allopathic medicine comes, that was the things they used to resort to get cured. So one of my friend advised, he put this one, you bear the pain so that it goes away permanently. Because it is for the, I don't know how it is.
No, that is remarkable, and regardless of how it happened, I'm glad it happened. Now
Yes,
the
sir.
question is whether it was just a chance event, whether it was you were anyway going to get cured, and around that time, and you unnecessarily subjected yourself to that pain of nimbu juice in your eyes,
Yes.
or whether it was directly because of that, we still do not know. And
Yes, sir.
And also why you had this type of allergic reaction. What was the cause? That also needs to be.
I don't know. Yes.
So there may be many things. that remain to be tested, remain to be questioned and examined. But
Yes, sir.
that's a good point to remember.
Yes, sir.
Thank you very much,
So,
sir.
one more question, sir.
Mhm.
Hello.
Yes.
Hello.
So the question
Yeah.
is if there are any drivers or check for ethics in research.
Yeah, yes.
It is
There's a check for ethics in the
no,
research.
it's not self-regulatory.
Mhm.
It is not self-regulatory. It is there is a lot of regulations around ethics.
Yeah.
Before we do anything in any research, we have to provide a plan of research, proposed research, and get it approved. And funding is then, determines whether or not that research will be done. And then even if it is not funded, but you still want to somehow do one particular experiment, before you do it, you have to get the approval from the university where you are working. And the universities have this fail-proof mechanism where what they do is any research done in that particular university, they have to give assurance to higher funding agencies or government agencies that they are all done in an ethical manner. If that assurance is broken, the university does not get a single penny from the government. So there is a very strict requirement for the universities to ensure that the ethical requirements are met. And these are all guidelines that are provided from top in the federal government or central government level. There are agencies which give you guidelines and policies, and we have to follow them.
Okay, sir. So
One more question.
Causes
Yeah.
of eye cancer are again many.
treatable or not?
Most important one is sun exposure. People who are living in high sun index, solar index areas where there is high altitudes or in the tropics, they are outdoor laborers. So it's definitely one reason. The other is genetic causes can be there. So cancer, like any other part of the body, is totally unpredictable. Cells have to divide, and we all are products from a single cell in the embryonic stage, and we have come to this stage where billions of cells are there and functioning together. If some regulation goes awry, you have problems such as cancer in many parts, and most of the times the epithelial cells are the ones which go through this type of a problem. And so for the eye specifically, sun exposure and some viral infections can be the source also.
Okay. I think we are running short of the time right now. So we do have the parallel sessions, and also at the same time, it's very late night for you, sir. So we will.
It's actually early morning already. Swami, it is at 12:33 in Pittsburgh.
Yeah. So
No, but
now
it was a
I
pleasure.
think it's the right time to wind up this particular session with a note that the university really compliments for your great pleasure and providing your valuable presentation, sir. That's very excellent presentation, which is an eye-opening for our students, especially with the science and society. What all the different parameters are the things that which can be able to focus it upon. Really, we are overwhelmed by your talk and whatever the inputs that what you have provided, sir. On behalf of the directorate and university, I really thank you for the participation, sir. And at the same time, I also thank all the audience who have been live streamed in different parts. Also, so who are there in the offline, and because they have been live streamed in certain departments. So they are also, thank you very much for offering that particular thing. So I think with this note, we will close this particular session, sir. Thank you very much, sir, for participating.
Oh, thank you, Swami, once again. It was a very good talk. And I wish you all the success in the days to come. And a lot of work can happen.
And also request you, sir, whenever that you are there at India, please drop into US, Bangalore, so your place, so that we can have a discussion with regarding.
Definitely.
Thank you, sir.
So thank you, sir.
Thank you, Dr. Swami, Dr. Raja here, and my wishes to Dr. Sudha as well. Bye.
Please give my best wishes to all postgraduate students. It is their week. They have been working hard and achieving, all good results. And they're the ones budding scientists who will have bright future. So my best wishes to them, and they can, whenever they have any question, if they have anything that they think that I may be able to help, I'll be glad to. That is the least, as an alumnus, that is the least I can do for the University of Agricultural Sciences. I'll be looking forward to situations where younger scientists from there come up and make wonderful progress in our field. Thanks
Thank you,
again.
sir. Thank you.
Thank you, sir.
Thank you. Bye.